Hello, everybody. I hope you can hear me and see me all right. My name's Giles Whittell. I'm an editor at Tortoise, and this is the weekly Tortoise ThinkIn that we call Sensemaker Live. And I'll explain very briefly in a moment what all those words mean. But the reason it's going to be brief is that we're lucky to have with us today Professor Helen McShane from the Jenner Institute at Oxford, but only for the first half hour. And so I do want to get into chat with Helen as soon as possible, because we've got a lot of ground to cover on our main subject today, which is COVID vaccines, the race to produce them, whether we're getting over optimistic about timings on that. For those of you, and I suspect there aren't many, who have not been to a ThinkIn before or an electronic one, it is the core of our slow news at ThinkIn. It's where we try to slow down, wise up with constructive thinking and listening to each other on important subjects. Sensemaker is what I and my colleagues Claudia Williams and Barney McIntyre edit and write every day of the week. And that's what we use at Tortoise to make sure that we don't miss anything, so that those of our members who are news junkies get their fix. And this, on a Friday lunchtime, is where it all comes together. We take a step back and, as I say, try to focus on what we think is really important. And there's absolutely no question, as my other colleague, Chris Cook, has pointed out more than once, that the search for a viable vaccine as a plausible exit route from the lockdown that we're all in is the big story of the next few months, and possibly longer than that. We also have with us today other people from the front line, as it were, including a volunteer in the Oxford trial, Ian Sharp, and we'll get to you, Ian. Thanks very much for joining us. I hope your arm isn't too sore. I'm fascinated to ask you a bit about that. But first, Helen, you are, a bit of housekeeping. This lasts an hour. We do record it. That doesn't mean that we'll quote anybody who doesn't want to be quoted. We just produce a roundup of ThinkIns at the end of the week called Heard It at ThinkIn, which you can see on our app. At the bottom of your screens, you'll see you have a list of participants you can click on. You have a chat button. Do join in the chat. You can see Claudia has welcomed everybody there, and I will be asking Claudia to make sure that we don't miss important stuff that is put up there. And really importantly, when you click on the participants list, at the bottom right-hand corner of your screen, you should see a grey button which says raise hand. A really important part of this process is that anybody who wants to should feel able to pitch in. It's all based on an old-fashioned newspaper editorial conference, and you can do that by clicking on that button to raise your hand. Let me start by asking anybody who thinks they know the difference between the Oxford vaccine trial that has recently started and other vaccine trials that are going on in the world to raise their digital hand and see if we get any blue hands. Helen, this is not for you. I'm coming to you in a second. Okay, that's very brave of you. Mehdi, I'm going to ask you, what do you think the difference is? And this is, I'm not being a know-all here, I don't know it, between the Oxford vaccine trial, if we use that shorthand, and others.
Okay, I'm taking a wild guess here, but I believe it's actually being tested on monkeys, on animals, live animals. Could you hear me?
Yeah, so I could hear you. So you said you believed it was being tested on monkeys. Giles?
Now, I can't hear Mehdi. Can everybody hear me? Barney, could you wave if you hear me?
I can hear you, Giles, and I could hear that question or that comment.
Okay. Well, let's then go to you, Helen, and please tell me if my connection becomes unstable. Let me ask you, first of all, what is the difference between the Oxford trial and the others, including, for example, the Moderna one, which started even sooner in Seattle?
Yeah, so I think, I mean, in a sense, there is no difference in the way the vaccines are tested. We have an extremely straightforward vaccine testing pipeline, development pipeline, that everyone who develops vaccines, whether that's in academia or in industry, follow. So to start with, you develop a vaccine, and I can talk about that in a minute. You then test it, usually in animal models, to see, firstly, is it safe? Secondly, to see, is it stimulating what you think is the right kind of immune response? And often what you do in those animals as well is you test what we call efficacy. So does it actually work? And you test that by infecting the animals with whatever infection you're trying to prevent. And then you see if the animals get the infection compared to those who haven't had the vaccine. And those animal studies form the prelude to then moving things into humans, where you do exactly the same things. You start with small scale studies to test safety. Are vaccines safe? You then move on to look at the immune response the vaccine is inducing. Is it what you think is the right kind of immune response for this pathogen, this bug? And you then move on to test efficacy. Does it actually work? And to do that, we have to do what we call randomized controlled trials, where you have a large group of subjects, half of whom get your vaccine and half of whom get a placebo, and then you count the number of cases in each of those arms. And obviously, if your vaccine works, you have significantly less cases or people with the disease in the vaccine arm compared to the control arm. So in that sense, the trials are no different in Oxford from anywhere else in the world. What is different is there are a variety of different vaccine platforms being developed here. And I think that's really good, because we don't know what the best kind of vaccine is going to be here. We do know there are a wide range of ways to make vaccines. Giles, stop me if I'm talking too long and you want to go more to questions, but
I want to jump in with one specific one, which is the Oxford one in any way an RNA/DNA vaccine? And am I right that none of those, although they have certain advantages in terms of speed of development, have ever been brought to market?
Okay, so no, the Oxford vaccine, strictly speaking, is not a DNA or an RNA vaccine. And you are correct that there are no licensed RNA vaccines at the moment. There is huge potential to RNA vaccine technology, but it is as yet unproven. So the vaccine that we're developing in Oxford is a different kind of vaccine. I mean, broadly speaking, vaccines can be split into two types. Either you take the whole, what we call organism or pathogen, the whole bug, and you kill it, or you, what we call attenuate it, so you make it weaker. And so I work on tuberculosis, and BCG, the TB vaccine, is an example of a whole organism vaccine that has been made weaker. The other way to do it is to take one or a few proteins from that pathogen, that bug in question, and then couple that with what we call an antigen or a protein delivery system to deliver that bit of the bug, if you like, to the immune system. Now, you have to do that with something to stimulate the immune response. If you just gave that protein, it would typically induce a very strong response. And there are two ways you can couple it with something to give a strong response. The first is you give it with what we call an adjuvant, which is a way of stimulating the host immune response. The other way is you develop, and this is what we're doing in Oxford, a recombinant virus. So you take a virus that is weakened, that we know is safe, and we're using a virus called adenovirus that we've used for a lot of different platforms and have a lot of experience with. And you clone that protein into the virus. So then when you give this adenovirus, it's got a protein from coronavirus in it. And then when your immune response sees it, it sees the adenovirus, which stimulates the immune response, but it stimulates an immune response to the coronavirus bit. Does that make sense?
Yes, insofar as with my O-level science, I'm ever going to understand it. Let me ask a question that I think will be on a lot of people's minds, prompted by a very detailed piece in yesterday's New York Times. I think was intended as a reality check. How is it that your colleague, Professor Sarah Gilbert, can express sort of 80% confidence in a good outcome from the trial that's just begun, when 90% of vaccines that get to this stage don't work?
So we know that this vaccine technology that we're using, recombinant viral vectors, recombinant adenoviruses, are a very good way to stimulate an immune response. And we have a reasonable expectation that the protective immune response that's going to be required is going to be stimulated by this vaccine. That will largely be an antibody response. And to be honest, vaccines that stimulate antibodies are a lot easier and more straightforward than vaccines that stimulate what we call T cells, the other arm of the immune system. Now, it may well be that we need T cells as well here, but we know that this platform stimulates T cells and antibodies. So we are comfortable that the platform itself works, in that we've used it for malaria, we've used it for TB, we've used it for HIV. So we're confident in the platform. We've identified an antigen from the coronavirus, and it's been cloned in, and we are now testing to see whether, firstly, is it safe, and secondly, whether it stimulates the immune response we are expecting it to stimulate. But because of that prior experience with this platform, we can be reasonably confident that it will stimulate an immune response. The key question will be, does it protect? Does it stop people getting coronavirus? And of course, that's the only important question. We won't know that unless we can test it in efficacy, unless we can randomize a large number of people to this vaccine and see how many people get coronavirus in the vaccine arm versus the control arm. That is the only way we'll be able to show if it works.
That there was part of it there that I hadn't twigged, and that's really interesting, that the platform has worked with malaria, TB, and HIV already. And
well,
this
when I say it worked, we know that it induces immune responses. Yes,
right.
I mean, they're not licensed vaccines, but we've got a lot of experience all the way through to phase 3 clinical trials in Africa. So we know these platforms work.
And this week we found out that in a small but significant trial using monkeys in the States, which is a part of your effort, part of the Jenner Institute effort, monkeys treated with the vaccine did not get sick with COVID. Now, how significant is that, given that we know from reading the newspapers all these years that there is sort of limited crossover from trials with monkeys, even if they're the closest thing that there is to human subjects to humans? So how significant was that news to you?
It's an extremely important result. You're absolutely right. We don't know how predictive animal models are of human responses. The only way we can test, or the only way we know if vaccines work in humans, is to test them in humans. However, NHPs or monkeys, non-human primates, are widely felt to be the closest model to humans in terms of biological plausibility and in terms of predicting a response in humans. And that NHP response is extremely significant. There's a very clear effect there. And that gives us a great deal of confidence that not only was the vaccine completely safe, but it actually did something. It actually stopped those monkeys getting infected. That is an enormously important result.
So if it's safe for monkeys, we can be reasonably confident that it's safe for humans?
In as far as that model represents human disease, yes, right. Absolutely. I mean, we can't be 100% confident until we test it in humans. That is why we do the human trials so carefully. And the Jenner Institute over the last 25 years has been extremely good at developing vaccines for a whole wide range of pathogens and testing them very carefully in a very highly controlled setting in small numbers of subjects to start with, to absolutely ensure that vaccines are safe before we roll out larger trials.
Now, Katie, my colleague and Tortoise's co-founder, is pointing out or asking in the chat about this business of needing a certain prevalence of the virus in society for the trial to work. Is that going to be a problem? And by the way, anybody who has a question for Helen, we normally have a no questions rule because we want sort of statements and want to hear points of view more than questions. But I hereby waive that. We've got a quarter of an hour left. If you have questions for Helen or just something you want to say, do please raise your electronic hand. But in the meantime, is that going to be a problem? If the infection curve is coming down, will there not be enough virus in the community to make this work?
It's obviously fantastic that the infection curve is coming down, and clearly that is an enormous public health success. But you are right, you need a certain level of disease in order to be able to test if a vaccine actually works. So you need a certain level of transmission, because if there isn't enough around, then you don't get statistical significance of a result. And it's absolutely critical that we get a clear, statistically significant result. I mean, we have plans to start to test this vaccine once we have tested its safety and its immunogenicity in other countries around the world. And it may be that other countries have a higher burden of disease, and you know, those plans are not firmed up yet. And I'm not part of the immediate development group, but you know, so it may be that we can still test the vaccine, but just not in the UK, but can still get that efficacy signal.
Are you thinking of anywhere in particular?
I think it's too early to say at the moment.
Right, but presumably you're looking quite carefully at countries where they might be on the beginning of an infection surge.
Exactly. So, for example, I mean, we have a lot of experience with working in trials in Africa. And in fact, you know, the African subcontinent is somewhere where, you know, this disease is a real worry, because of course they don't have the infrastructure that we have in the developed world, right, and where a vaccine is even more urgently needed. So it may be that trials are more feasible there. And again, we have a long-standing history in the Institute of being able to test vaccines with academic collaborators on site, on the ground there, to do really high quality, carefully conducted trials in those kinds of sites.
While we're on Africa, my colleague Basha did a podcast this week on the looming crisis expected by healthcare workers in the DRC, the Democratic Republic of Congo. At the same time, David Pilling wrote in the FT that possibly, just possibly, some sub-Saharan African countries might have dodged a bullet. What's your, and you can see so far very low reported infection rates compared with Europe in, say, South Africa, big and relatively developed economy. What's your take on whether the worst is yet to come, or in that part of the world, or whether in fact the worst there might not be as bad as it has been here?
I think it's very difficult to say. I mean, I've spent quite a lot of my life working in South Africa. You know, if this epidemic or this pandemic takes off in South Africa or other African countries, it will have a devastating effect on their economy and on their very, very fragile healthcare system. Difficult to say whether the relatively low number of cases, and actually in South Africa they are going up, is because of limited testing and limited infrastructure for testing and healthcare, or whether, you know, often in these countries there is a bit more geographical separation and, if you like, social isolation than there is in the West anyway. I think it's too early to say.
Now, Keith is asking in the chat about the timing here, and this is something that really struck me from the New York Times piece I mentioned earlier. Vaccine development can take decades. You've been working on particular class of vaccines since the beginning of this century in South Africa. Some have taken 50 years to develop. We're talking about the most optimistic prognoses for this trial, and one in the States, are for an experimental vaccine to be used on an emergency basis by this autumn. But even the most optimistic forecast that I've seen otherwise are for 18 months. But even that is an incredibly accelerated time frame, isn't it?
It is. But one of the things, and the same thing happened in the Ebola epidemic, you know, one of the things that happens when there are global emergencies like this is people move as quickly as is humanly possible. And it's absolutely clear across the globe that people are moving as fast as they possibly can. Approvals for trials, approvals for manufacturing that normally take weeks, months, happen in days or hours. And this isn't in any sense because corners are being cut. No one is cutting any corners here, but people just understand there's a global emergency and every single human resource possible is put on to moving things as quickly as possible. So that's fantastic. And of course, it gives one a sense of what one can do when there really is an emergency, although I don't think one can live one's life like this all the time. So that's important. I think it's important to see there are different factors here. So the first thing is, you know, we have to test to see if the vaccine is safe. That's clearly critical, and we have to do that carefully. We then have to test to see if it's stimulating what we think is the immune response we need. And ultimately, we have to test if it works. That, as we've already discussed, will depend how quickly we can test if it works, will depend on how many cases there are and what the prevalence is at any one time. In parallel with that, and I think again, often these things are done in serial rather than in parallel. Normally one gets efficacy and then moves on to large scale manufacturing. What's happening now is people are saying, okay, we have to do this at risk because this is a global emergency. We have to make plans to manufacture huge numbers of doses as quickly as we can in parallel with doing that clinical development. Now that is at risk. It's risking using money that, you know, if the vaccine doesn't work, we shouldn't have done all that manufacturing, but clearly it will be a terrible place to be to get a signal that the vaccine works and not have then supplies to roll out. So there's a huge amount of effort in Oxford and in all the other academic and industrial groups developing vaccines to run these things in parallel.
Right, thank you. By the way, Ian, I think we've got Helen for another few minutes, and if as a participant in the trial you wanted to ask her anything, please do. In the meantime, I've been prompted to ask, and this is clearly a good question, let's assume everything goes well, and bearing in mind the obstacles to mass production, what is your best estimate of when we might have, all being well, a wide availability of a vaccine in this country?
That's an extremely difficult question. There are many, many factors that feed into the answer to that, to do with how quickly we can manufacture, how many global manufacturers we can get on board to help with that, how quickly we can do that efficacy testing, and that will depend on the prevalence of disease, which is, you know, unclear and uncertain, partly because of problems with testing and partly because of problems with what you actually use in those denominators to make those calculations. You asked the question about widespread use in the UK. You know, that is not going to be this year, widespread use in the UK. You know, if we get to the end of the year and we have an efficacy signal and we have manufacturing being ramped up, that would be fantastic. But, you know, global use we're not going to have by the end of this year.
Right. And Keith is asking in the chat, a slightly separate but obviously related question, what's your estimate of the prevalence, let's start with the UK but also sub-Saharan Africa, of the virus, because the level of testing we've done gives no indication at the moment, does it?
So I don't have an estimate. I'm no better than anyone else on that. I mean, I think it's incredibly difficult. You know, if we'd been doing wide scale testing, we will be able to make a better estimate of it. It depends whether you use the hospital admissions, whether you use, you know, ITUs. You know, I think there is some important work going on. There's a serological survey going on, being led by Sarah Walker, one of my colleagues here in Oxford. I think that will give us the kind of information we need to be able to estimate prevalence. But in the absence of that data, I can't make an estimate any more than anyone else can.
And if we had been doing more testing, would the trial, would the data that you're getting even in an early stage from the trial be more significant?
No, because the data we're getting from the early scale trials is it safe? And as I say, we've got a lot of experience with this vector. We expect it to be safe, but clearly we have to do those trials carefully. And I'm very grateful to people like Ian who volunteer with fully informed consent to take part in these studies, and we get a sense of what kind of immune response it's inducing. That would not be altered by having a better estimate of the prevalence at the moment.
Okay, people are using the chat to ask their questions rather than the raised hand, which is fine. I will pass them on, and we'll get through them as quickly as possible. Lara is asking if all goes well, who would get how will the vaccine be rolled out, which countries and who in them would get it first? Any idea?
We haven't worked those things out yet. Those are not decisions that I will be taking, and clearly, there will clearly be a needs basis, both in terms of who gets it within a country and which countries get it. We have to give this to the people most in need. That might be healthcare workers, that might be vulnerable people, that might be people who have immunocompromised reasons why they will be more susceptible.
Okay, thanks. Clearly a tough one. Katie asks, the way things are going, how are you going to live your life in the next few months? I hope you can still hear me.
To this disease. I can still hear you. Well, I think I need to be clear. So I work in the Jenner Institute alongside my colleagues, Adrian, Sarah, and Andy, who are leading these trials and this vaccine development. I'm not personally working on this vaccine. In fact, I'm personally coordinating drug testing trials in Oxford at the moment. Meanwhile, I will continue to work on a tuberculosis vaccine, which is what I do as my daytime job.
How long is your working day?
Long.
How long?
Very long at the moment, right. 12, 14 hours.
When you say you're coordinating drug trials in Oxford, is that COVID-related drug trials?
Yes, absolutely.
Well, it's a shame that it's 28 minutes past, but before we've got to that, because I'm fascinated by whether we should be as optimistic as some people suggest about stories about remdesivir and other drugs that are already out there.
Well, there are a lot of drugs being tested in trials. I think we've got, some preliminary results from small scale studies, but actually we need the results of bigger, better trials. I think, with all of this, with vaccines and with drug testing, it is important that we make sure we still get the same quality of evidence that we would expect outside of a pandemic. Because whilst there, of course, is desperation and, of course, is urgent need to test as many things as quickly as possible, cannot lower the bar for the quality of evidence. The drugs that are being tested have people that need those drugs anyway for other reasons, and therefore if we gave them to people for COVID, perhaps on flimsy evidence, we might compromise the supply of those drugs to the people who need them for other reasons.
And you're talking about specifically?
I'm talking in general. I'm talking in absolutely.
But you're an expert on BCG, and you were mentioning beforehand that's been talked of as a potential therapy here.
You're right, and thank you for that. There is no evidence at the moment that BCG protects against COVID. There is a long body of somewhat controversial literature that BCG may have protective effects against other infections. It is being tested in 2 large-scale clinical trials. We must wait for the results of those trials. The world that needs BCG protected against TB should not have their supply compromised.
Right, well, thank you. Helen, it is past. Thank you so much for your time. It's been really interesting, and thanks everybody for pitching in with questions. I think you have another call to get to, so I will let you go. Thanks so much for joining us.
Pleasure. Bye-bye.
Bye. Ian, thank you for joining us as well, and thank you for your patience. Now, tell us a bit. We're all completely fascinated as punters or potential punters. First of all, why did you decide to volunteer for the trial?
I think, firstly, it's sort of right time and right place. I happen to live in Oxford, and I was trying to recall when I even first heard about this trial going on, and I can't think where I came across it. But I made an application on their website and didn't expect to hear anything back. And to my surprise, a week later, I got accepted to do an initial screening, which is just a first step to determine whether you're fit enough and look at your medical history. So, I mean, I do feel it's just really sort of fortunate to even have the opportunity. But as to why, I think it's multi parts to that. I think the first thing, like a lot of people that may find themselves, as I do, working from home sort of day in, day out, you feel like you would like to have an opportunity to contribute, and everyone can contribute in different ways. I think when this opportunity hit me, I thought it was sort of a golden opportunity for me to contribute in a way that could end up being extremely significant. Whether it turns out to be a success or not, it's more about contributing to the learning process of science, so people can learn a little bit more. So it has the potential to be a very valuable contribution, and myself and hundreds of other people just had the opportunity that I didn't want.
And what's it actually involved for you so far? I was reading on the university's, the General Institute's website, that you're required to take notes and things.
Yes.
And you had the jab yesterday. Was it your first?
Yes. So I did the screening about 2 weeks ago, and they check all your medical records, etc., just to see if you're fit. There is certain criteria that you need to meet. One of those criteria is that you can't have any symptoms whatsoever. As an interesting fact about that, I might want to share with you, that you can't have had any symptoms previously before taking the trial, and if at any time up until the inoculation you do, then you have to let them know, and you'll probably be excluded. So it starts out with that screening, and then I had got accepted, again, somewhat to my surprise, because I know it was very oversubscribed. Just for people's information, the first round of people was 500, so there's 500 of us. I think they're only about halfway through about 500 as we speak, I think. And I had my jab yesterday afternoon about 5 o'clock. And again, Helen did mention it, but just to make it very clear, it's 50-50. I have no idea whether I've had the vaccine or whether I've had a placebo, which actually is another vaccine. It's not just
right.
Sort of. It's not. Interestingly, the placebo is being designed to give the same side effects or potentially give the same side effects as the actual vaccine. So whether you have side effects or not gives you no clue as to whether you had the vaccine or not. And even the person injecting you doesn't know.
Right.
So obviously,
are you feeling any side effects, by the way?
Has the interest zero, absolutely nothing. And you're right to say that as of day zero was yesterday for me. There's a very comprehensive questionnaire that everyone has to fill out on a daily basis, same time, same questions, that go through very carefully how you're feeling, any symptoms whatsoever, share symptoms, whether you think it's related to the injection or not. So you have to share absolutely everything you might be feeling. The severity of all your symptoms, you have to take a temperature test and give them the highest temperature. If you think you've got any raise in temperature, you give that. You have to take measurements of the injection site and let them know any swelling, any tenderness, anything. So it doesn't actually take too long to fill in, but it's obviously given them very detailed feedback on a daily basis.
And one more question, and then I want to see if we can find Amy Peters, who's joined us here from Johnson & Johnson. But did they give you any advice as to how to expose yourself to the virus? Presumably, if part of the point is to see if this works, they would ask you to go out and about and get on a bus.
No, on the contrary, actually, they said, again, because they don't know, or most individuals don't know if you've had it. The advice, I would say strong advice, is carry on and I'm pretty much in lockdown. I only go out, maybe for, a little exercise on my own, or, visit to the shops. But the advice is to assume nothing. And I'm still maybe susceptible to get it. And that's not a good idea. So the opposite. I think, again, I'm always a bit nervous about sharing technical details, but as I understand it, I then have monthly for the next 6 months. I have to go back in and have blood tests and more tests, and I'm assuming they're looking for production of antibodies or whatever these particular cells are that they're hoping to be produced. So absolutely can't expose myself in any way.
It's really interesting. Well, look, it turns out that we have somebody else who volunteered for the trial, and we'll dive. Will, I'd like to come to you in a bit. We'll dive back into the nitty gritty. But first, let's take a step back, and I want to go to Amy, if we can. Amy Peters.
Hi, yes, how are you?
I'm well, thank you so much for joining us.
Sure.
Are you in this country, by the way?
I am indeed, yes,
Right.
although American by birth.
Right, as is most of my family. It's great to have you.
Thank you.
Now, Johnson & Johnson, as an enormous organization, as I understand it, is closely involved in all these endeavors in a separate vaccine development effort in the States, and also was widely reported to be involved in trying to ramp up production of equipment, ventilators as well. Am I right?
Yes, absolutely. So we at Johnson & Johnson, we are currently working on a vaccine. We have one primary candidate and then 2 backup candidates, which are sort of on track for clinical trials in September, with a look for it to be made available early in 2021. So it's kind of a case of watch this space. I know they are working fast and furiously with the idea that they have committed $1 billion US to then providing the vaccine globally at pretty much a cost basis. So very affordable, really ready to roll out. We've got the capabilities to mass manufacture and get it up and running.
And do you know the primary one, is that one of the newfangled RNA/DNA ones, or is it a more conventional vaccine where you're actually injecting an attenuated version of the virus?
I'm afraid I actually don't know that information, but I could find out and share it with you post meeting. Okay,
that would be great. I'd love to know, because because of the point that I raised with Helen earlier on, which is I feel that we should be skeptical about the very latest versions of this research, just because it's untested. I wanted to ask a general question, Amy.
Sure.
Is there a worry that in this race there is not enough collaboration between the participants, and as a result we end up missing the prize?
That's a really good question. I guess from my point of view, one of the great things about J&J as well is that we do try to collaborate with as many people as possible. So I know that there have been globally, many collaborations that are going on. The scientists in the scientist community, have been really positive about working together with this. And I guess really from my own personal point of view, I think everyone is really hoping that we will have a really good outcome, and everyone has been very open to collaborating. So I think the priority really is to get something out there, make sure that it's safe and that's usable, and that we can, protect people with the virus or against the virus. And I think collaboration is a huge part of that.
Clearly, at the manufacturing level, my understanding is that once somebody gets there on the science front, they will manufacturers all over the world will make their facilities available. But is Johnson & Johnson collaborating in terms of fundamental research at the moment with others?
As far as I'm aware, yes, and I know that we also are collaborating on the production front too. I will have to go back and double check who that is all with and get some more information. But in general, yes, we have facilities and capabilities to provide vaccine.
Right. Thank you very much.
Sure.
Will, you've raised your hand. I love a blue hand there. And you've also started explaining in the chat your experience with the trial. Can you tell us about it?
Yeah, so I'm a healthcare worker in ICU. And we got a message saying Bristol have opened up a centre to cooperate in the Oxford trial, and they were recruiting specifically healthcare workers. And I think that's probably the point that with the R number, I guess healthcare workers are going to be more exposed to the virus.
Right.
So be it appropriate vaccine participants. So I went through the screening and all of that. Much like Ian was saying, we're probably a few weeks behind where he is in the stage of the vaccine trial. So answered all the questions, as I said, asymptomatic, etc. But got the blood results back, and they were slightly abnormal. Not massively, just the hemoglobin was a little bit low, and the white cells. But so I've had to pull out of the trial. But I guess it brings up the point on vaccine trials that they just test these on all healthy individuals, where they won't be giving the actual vaccine to healthy individuals, if you see what I mean. So
will they not? I thought that the point was that they would be. My parents, both in their 80s, certainly are holed up at home, healthy, thank goodness for the moment, waiting for a vaccine so that they stay healthy.
I guess the vaccine, they're giving it to 18 to 55-year-olds with no other comorbidities, technically, etc., etc. So even though you know the safety profile in those people, you might be giving it to people who don't fit those demographics. So you won't know if it is safe or not in that, if you see what I mean. Right.
And in your note on the chat, you talk about a recovery trial. And I don't know what that is. What is that?
So that's another, that is looking at the different remdesivir, hydroxychloroquine. I think there's 4 or 5 arms to the recovery trial, and any patient in hospital who's suspected COVID or COVID can be recruited to that trial and be given one of those treatments, and 10 days of that treatment. All the data is put into a national database, and they're going to see, I think they'll be publishing some interim results soon on which of the 5 treatment arms, the results from the 5 treatment arms, and see if any one of those is superior. I think the treatment arms are no treatment, steroids, hydroxychloroquine, remdesivir, and I can't remember, I think it's inhaled interferon's the last one. So
one of those is Donald Trump's favorite.
Yeah, I guess so. But again, I mean, we don't know the evidence on whether it works or not in COVID. So.
Who's running that trial?
So I think, again, that's come out of Oxford. But, I mean, there'll be hundreds of hospitals around the country recruiting to that trial. But from just looking at these patients in the ICU, it's not that clear if one's more superior to another. Right. But I guess you need to look at all the data from thousands of patients and see whether one of them is statistically significant or not. And then we'll have an answer in the UK. And if one is better, I guess they'll use it.
Will, thank you. You say you're a healthcare worker. You work in a hospital in Bristol or what?
Yeah, in Swindon. I work in the ICU there, just a junior doctor.
Blimey. How are your days at the moment?
Yeah, they're all right. We do 3 days on and 3 days off, and they're 13-hour days. So it's just very bizarre. It's so different. Every single patient has COVID. And even though the numbers are dying off in the hospital, in ICU, they're in for a minimum of 10 days intubated and ventilated. So we're about 2 weeks behind the A&E numbers, because they might be coming in and out of A&E, but the sick ones that come to ITU tend to be intubated and ventilated for a good 10 days to 2 weeks. So we're still pretty busy. And then the PPE is just knackering. It just, you know.
Have you got enough of it?
By the end of the day. Yeah, to be fair, I feel like we do. And weirdly, even though you might be most exposed in ITU and they're the sickest, we have the best PPE. Whereas I feel my colleagues on the wards are at much more risk than we are in the ICU.
Quite a lot has been written in the past week or so about how, in fact, worst fears about ICUs becoming overwhelmed have not been realised, so that there is, even in London hospitals, a bit of spare capacity, hence the relatively underused Nightingale Hospital. What's your experience in Swindon, just in terms of beds used?
Yeah, so I think we're a 12-bed ITU, eight ITU beds in normal time. And they were told to expect, increased capacity to 40 to 50, which they've provisionally planned, and then the Nightingale for extra hospitals in Bristol. We've had, I think, at a maximum of 20 ITU beds at once, so didn't really hit that maximum of, I think, 30 or 40. So yeah, not as bad as they thought. But it's all the other things. It's not just beds. It's nursing numbers are probably our main limiting factor, ITU nurses. But then even like things you never think about, oxygen supply to the hospital. When they started looking at it, they could only supply enough safely for 30 ITU beds because the oxygen capacity to all the beds, that was the max, if you see what I mean. So yeah, it's not as bad numbers-wise as they thought it was going to be.
Do you think that's partly because people are simply opting not to go to hospital or being discouraged from going to hospital, especially if they're at care homes?
Yeah, it's hard to know. No, I just think the isolation has worked. And I do think the delay we had from mainland Europe's probably helped us prepare a bit. But yeah, thankfully it's not been as bad. And I do think the Southwest isn't as bad as there's a bit more space. We're not as dense as London, so probably haven't felt the effects quite as badly. Yeah.
Will, thank you for coming on, making it sound like a radio show. It's not a radio show. Mehdi is asking, do you reuse PPE?
Yes, well, they've started to. So the usually single-proof theatre gowns, they're now washing up to 3 times, but I feel that's okay, to be honest. I feel that's acceptable, but we're not reusing gloves, and I actually feel we have adequate PPE. I can't complain at all.
Right. So. Fascinating. Well, look, if anybody else wants to ask Will or Ian anything, do raise your hand. I wanted to come back to Amy briefly because Katie in the chat. Why don't we go to Katie? Katie's not backward about coming forward, and you asked your question about whether J&J will manufacture for others.
You answered it. You said that you knew that people would manufacture.
Well, I'm not honestly sure. Go ahead.
Okay. So I mean, Amy, I think you touched on it, which is that there's a lot of need to collaborate. But I was just interested that obviously, Helen and the team that she was talking about at Oxford, if they're lucky enough to get a vaccine that works, I'm assuming that obviously J&J would be happy to manufacture their vaccine if they found one. I mean, that was my question, really.
Amy, just hold on a second. I'm not hearing you.
Oh, apologies. I think from a J&J point of view, I think they would be very open to, of course, discussing that. I can't say for sure, what they would actually do because I'm not in that current environment, in that current situation. But I definitely would say that they would be very open to that. And in fact, I did find the collaboration agreement that I had spoken to earlier. It's with Emergent Biosolutions, and they are going to be doing some integrated vaccine manufacturing and supporting, our product supply for 1 billion doses of the vaccine itself. So I would imagine they would be very open to it, but it would be a discussion for someone way above my pay grade.
By the way, Amy, I was reading just before we started a piece on the CNN website about a meeting that would have been fascinating to be at as a fly on the wall in the White House with Trump and representatives of all the big manufacturers and some of the small ones, at which this relatively small outfit, Moderna, said that they could get an experimental vaccine out within weeks rather than months. And this was immediately latched on by the administration, and nearly $500 million was shunted their way to accelerate it. And this is one of the unproven RNA, DNA ones. Have you picked up any sense, let's make this off the record, in as far as it can be. Have you picked up any sense sort of within your own organization as to whether the administration in Washington is making the right bets? I mean, I know you as a big established player have also received large chunks of public money and good luck with it. But is there a concern that in the rush to get the prize. Too much is being bet on unproven organizations and unproven sort of approaches?
I'm sure that you can appreciate. I'm actually here in the UK, and so I have very little visibility about what is happening in the US and in terms of anything that's going on in the White House. So I'm afraid I can't answer that question for you.
Oh, that's fine. I just thought there might be some water cooler scuttlebutt, but actually it's difficult, isn't it? When we're all at home, there is no water cooler.
Absolutely.
Before we run out of time, I wanted to ask everybody, and this is something that I've seen the eagle-eyed Claudia actually asking you, everybody in the chat, or this is a variation on the theme. If the development time for a vaccine turns out to be more normal, and we don't have one until next year, certainly for large numbers of people, I'd be really interested to know if there are older people amongst the 80 who are on the call who are willing and planning to stick it out, like my parents are, until there is one, or whether you are living instead with some notional deadline or cutoff point you put in your head, or whether you're fed up already. Is there anybody who... Sue, let's go to Sue. What do you think about this?
I have no option as my daughter dictates what I do. So I've even stopped going out for a walk. It's quite difficult. And I could probably become an obese alcoholic at the end of this if I have to stay in my house.
So she's not letting you out at all? No.
She said to me, "Mother, you can't go out."
And you're going to abide by that even if it stretches on into next year, are you?
Possibly, yes.
Let me ask you.
I think that we have to. You have to use your brain on this, and I don't particularly want to become as ill as I know a lot of people have. Mm. Equally, I'm old enough to have seen quite bad outbreaks of things and probably had them and got over them, but I don't think anybody knows what this is, and that's the uncertain part about it all.
If this autumn you were offered a vaccine with the caveat that this has been produced on an accelerated timetable, we have done trials, but this is being offered to you on an emergency basis, and if it were not possible to give you an absolutely cast iron 100% guarantee that there would be no side effects, would you take it?
Yes.
Would there be a threshold at which you would not take it? We're talking hypothetically here, and it's foolish of me, in fact, because...
No, I don't think so. I think I'd offer it to my mother, who is 101 in a couple of days, and she's still going. Just don't catch her at breakfast time when you can't interrupt her. But I would say that the older the person, the better. If you want to see if we are particularly, I'm not convinced that we are the most vulnerable. And that's the one thing that I find is quite difficult to understand about why they are protecting or so-called protecting the over 75s or over 70s.
Because who are the most vulnerable? You mean people with pre-existing conditions?
Yes, but I mean, I'm on take 2 inhalers, but I wouldn't say that I am vulnerable to most things. I will get something that nobody else has got. Everybody goes, "Oh God, right, okay." But equally, I think that if we're going to get it, we're going to get it. If we're going to die, we're going to die. But that's my outlook. So yes, I would be a recipient for the injection.
Okay, well, thank you. Edmund, you've got your hand up.
Hey, Giles. I'm not of the sort of elderly generation, but hopefully I can speak for them. Although I am actually wearing a dressing gown inside because it is cold today. But my mum and dad, who are in their 60s, I don't keep them on the same leash that Katie does with her mum and Sue. Sue, I do hope you are getting outside as much as possible. But they would certainly say they will be hunkering down for as long as it takes if the vaccine is not coming, and they won't be venturing out. And I think if I was in that age bracket or was worried, more of an at-risk age, I would be hunkering down as well and being careful. But then it slightly comes to the point whether that's a sort of position of privilege or fortune. I was listening to Basher's Tortoise Slow News podcast yesterday, speaking to someone in DRC, and basically said that, if you are able to afford to stay in and not go out and earn a living, then that's fair enough. But if you're in a different sort of socioeconomic situation, then that's not an option, and that's where it will get difficult for people of a certain age or people across the country, and that's where you have to go out regardless of whether you feel you can or not.
I think that's very true. I also sensed in an earlier ThinkIn this week that elderly people up in Yorkshire with more space and less virus were more likely to get cross and say, the heck with it, I'm going for a walk, than people, for example, in London, where there's a very palpable sense of the virus being close by. Ian, before we run out of time, I wanted to come back to you because you've raised the question of travel. And is it the trial, your participation in the trial, that is preventing you traveling? And if so, how are you able, if it's an important part of your work, how are you able to keep doing it?
It feels like it's a bridge that's yet to be crossed. So I do, for my job, very frequently travel abroad. I don't think flights will be commonplace in the near future. But also aware, China, for example, insists any visitor has 14 days quarantine. And I think the problem's always going to be is how do you, keep things from, coming back again. And I think these quarantine between countries may be a part of how it's dealt with going forward. So I can see it being something like 14 days quarantine when you arrive, 14 days quarantine when you come back. I think that may be on the cards. So for me personally, and the business I work for at the moment are adapting to work remotely. So I think for short-term trips at least, I don't think it's going to affect me because I don't think anyone will be doing it.
Right. I mean, the idea of a business trip that would have been 1 day turning into 15 days is
exactly
extraordinary, isn't it? Claudia, you've been keeping a sharper eye on the chat than me. Is there important stuff that hasn't been aired? Apart from the fact that Ed thinks over 60s is old. Yeah, Ed.
That's the important thing that's been said. I've had a couple of different people emailing, privately messaging me, basically saying that they're finding this whole conversation about the vaccine really interesting, but as someone that doesn't necessarily understand the science behind it, they've had to put a lot of personal effort in to get to grips with it, which I think is really interesting. And a lot of people, a couple of people, there's one person who has said that they were a medical librarian, so they feel like they should understand it, but they're still having to put a lot of effort in. And I just think it's very interesting how much, people are really, really keen to understand this, but journalists who are writing about this all the time, there's so much to take on board and it's changing so quickly that it can be difficult to stay up to speed.
Well, I think journalistically, there's clearly two challenges. There's getting too deep into the weeds so you forget to tell the story. And there is the more serious one, I think, of getting swept up in too much optimism and finding that you have more or less effectively persuaded your readership that there's going to be a vaccine possibly as soon as September, when the chances of that are very, very small. And even if it works, it'll only be available to a small number of people. it is 2 o'clock, and I think we try to be very timely because everyone's got other things to do. There is a question that I want to take advantage of the fact that you're here. It's separate, but it's important because we've been getting we've spent a lot of time thinking about it at Tortoise this week. Please raise your electronic hand if you think it is too soon to be trying to figure out who is responsible for the fact that we have such a very, very high death toll in the UK. Okay, interesting. I'm looking at about perhaps a quarter to a third of us. Thank you. That's a really, it's a small sample, but we take the sample that is Tortoise membership very seriously. I want to thank Ian for joining us and for taking part in the trial. We really hope that it doesn't give you any nasty side effects and it does give the world a vaccine. And I think my sense is that we'll all be as patient as we can be. I want to thank Will. Thank you as well for your service, as they say in the States, but also for getting on the call. It's really interesting to hear from the belly of the beast that is Swindon ICU. And I want to thank everybody for joining the call. I've learned a lot about it. I'm sorry if some of the vaccine details went over people's heads, but this is our lives now, isn't it? We've all got to try and figure out what is promising and what isn't and why. So that's it for Sensemaker Live. I think one thing I didn't say at the beginning, so I'd better say now, is this has been brought to you by Santander, our founding partner, and we really are genuinely grateful for them for making the whole, for helping the whole Tortoise ship float. They believe in what we do, and we're grateful to them. And above all, to you for joining us. Let's have a little wave and have a great weekend. Thank you very much, and goodbye.
Thanks, everyone.